we construct the network from using our 14.
Sorry, I can't hear you very bad. The sound is very bad.
I change my headphone. Hello. Yeah. Now is
Yes. Second, that maybe it's better.
okay. So as you know, like in our previous meeting, we have constructed protein detection network. Now in this meeting, my voice is clear.
Yes. Now it's better. Okay, thank you.
So in this meeting, we will try to find functional module in our protein detection network. So we will use basically these 4 methods, including like DIAMOnD, MCODE, leading eigenvector, and Mtgo methodology. So first, like once we have start with we start with DIAMOnD, because DIAMOnD is it is a disease module detection method which uses 3 algorithms that are applied to find the functional module. Like one algorithm is like link community algorithm, second one is like Lovian method, and third one Markov cluster algorithm used. So this method is trained by like approximately diseases proteins. So once we have provided once we have provided the protein interaction network in the form of text file, then we can set the criteria, how much threshold, how much number of DIAMOnD proteins you want to find. So here, like so like once we have just provided the our query network and our query proteins. So basically we provided 2 input files here. First is like our network file, second one is our protein of interest. So like basically DIAMOnD is very useful for that time when we know that we have like several disease associated proteins. So we can find the interactor of these disease associated proteins and sub-interactor. So you can see that in the map, like here you can see in the map, like seed proteins are denoted by like a red circle ball, which is our proteins. Our protein means like these are the disease proteins in the network. So these are proteins are highlighted in our protein interaction network, and these are the crucial seeds. Crucial seed means that these our proteins are interacted with the, another protein, which is denoted as like crucial protein, and other DIAMOnD proteins are just which are predicted, not our seeded protein, are in a like orange color. So basically we can get the basic idea of like we can get the number of complex region which are interacted with our seed protein, and their first neighbor are important in terms of our functional module identification, because like proteins interacting with similar set of other proteins in a given networks. So basically also that have higher probability of sharing similar functions. So this protein may not directly interact, but they are indirectly working towards similar cellular functionalities, and that should belong to the same module. Once we have like predicted all these proteins, then we can highlight in our network, like DIAMOnD proteins are colored in red colors, and green nodes are those proteins which are interacted with our DIAMOnD proteins. So then we can go and find cluster and then go for pathway enrichment analysis of each cluster. So basically I just explained that how it works. So basically like input file, we need to see that this file contain disease associated protein, which we call like seed protein, and we did not. So okay, so in the DIAMOnD is basically run by the Python script, which we have provided 2 files, seed protein and like all complete protein interaction network. So we will we will like run in the coming time. So and second method is like leading eigenvector method. This is also this is also paired with the functional module in the protein interaction network. Basically eigenvector measure the influence of the particular node in the network. If some node has higher eigen values, that means that interacted protein also have a high eigenvalues. So what we do, like first we have just take our protein interaction network, just break into communities. So once we run that R script, that is show how much probability of this network is broken into communities like 2, 3, 4. So like we just name them like a level 1. So like level 0, we use our protein whole network, which we are going to find the functional module. Then we break this main network into sub network, and then some network also be network break into sub network and sub communities, sub communities in multiple level. So we break this community up to that level when the modularity of this network becomes 0. So basically, most of the time when we break the network, it goes up to 6 level, 7 level. So it's up to you. If you find your like functional module where that module contain your protein of interest, then we can stop the breaking the communities. So these are the also, but it is very long time taking method, which takes too much time, because we have to first run this main network into communities, then we have to take one by one sub networks and break again. So that is like a chain of the networks. Then we have to identify what are the communities are important for us, and what are the communities has our hub proteins. Then once we have like identified all these proteins communities, then we again go for like pathway enrichment analysis of each class, each communities. So that is another method, like MT Mtgo. That is like it is a Java based program, which is like we can install in Windows and Linux also. So it is search, it is search into it search two criteria. First is take information like topological information, which is like our network file, and it is also take information from biological term geoterms databases. So here we have a list of proteins and their associated biological terms. So it takes both the information and then predict the some functional module. So here we have to provide like a input file as a 2D matrix, which is like A connected B, like 1 to 1. So it is like a network file, and this is import gene ontology file. It is also like all the proteins which have associated geoterm. Approx you can download from here, like 20,000 proteins are there and their associated terms. Then we can run this Mtgo, and that give the output in the form of two files, like one file is module based QGO that represents the module and associated geoterm, and second is name term, that is also network file. So first, so basically both are the informative file. We can use this file into Cytoscape, and we can get the maximum information. So it can predict all these module, and we can again go for enrichment like analysis of each module. So this is the overall is introduction of this module method. So now we will go for one by one to run these methods. Any question is there?
I have a question.
Yep.
I previously worked a little bit with DIAMOnD, and I recall having had some problems with the version of the Python, so I had to adapt it to Python 3. Now, I mean, this is a more technical question, but you said that you have some Python scripts. Do you use Python 3 and you had no problems, or how is the?
Yeah, I think I have Python 3.10.
Okay,
So,
okay, that's great. That's great.
yeah.
Thank you.
So in starting, I also faced like problem like the network X module is not installed. Then I see some video, because I'm not expert in Python, so I just see some videos tutorial, then I install, but it's fine now working.
Okay.
Okay, so I start with I think Python DIAMOnD.
Okay.
Now my full screen is visible.
Yes.
Yeah, so these are the files. First I will show you. This is our PPI network file, which is 2D file, like protein interactor 1, interactor 2. So you have to be more careful that you have not put a space here. If you put a space, it will not run. It will give error. So that is like that. No space, only comma. And seed genes, like these are the seed genes, which are like, suppose we have 100 genes, 20, like 200 genes. So these are the disease associated proteins or genes. So one thing you should remember, that seed genes, this name should be same. Otherwise you have to change in the script, because it
Okay.
take from this formation. Okay, and PPI also the same name. So I will show you this. Yeah, these are the same script, small simple script. You have to just uh, retrieve this information from. So now we can go for like command prompt. Now we can here. I think. My command prompt screen is visible now.
Yes.
Yeah. So like our folder is in E drive. So like, so we have to remove single space like that. Sorry.
You have to change the folder maybe.
Yeah, I think so. Yes, second. Yeah. Okay. Now it's run. So at least it takes like 2 minutes or 1 minute. Now results have been saved in the same folder. Yeah. So these are the top 100 proteins which are present in our network and their diamond score. So higher diamond score means higher, lower diamond score means higher significance. So we can get all these 100 top 100 proteins. Like we provided approx 300 proteins. So it predict only the top 100 proteins from the overall, and it gives us some score. And these score also you can use in the Cytoscape for like make a graph using like those nodes are higher in the diamond score. We can make it like visualize in a like big ball or some. We can set as edges also. So this is the method we can predict. And this is the we can also predict like 1,000. You can set not like limited to 100. You can predict up if your network, suppose your network is like 20,000 proteins. So you at least predict 100,000 proteins as a diamond protein. And this file as you can use as a node file and edge file in the main PPI network to draw the network as shown in the as shown here. You can set the edges, you can set the seed proteins. So clear here? I'm clear.
Sorry?
I'm clear. So I can go
Yes.
next.
Yes,
Okay.
yes, okay.
So I will send all these script and file after this meeting. So you can try. So now I will start like a second method, like leading eigenvector method. But that is R script. So here you have to provide like a main network file, that is SIF file. Like this. SIF file like just 2D array, just save as a .sif in the network. Like interactor A, interactor B. So that is called SIF file. So in we use igraph package and mass package to do this experiment to find this. So first, like we have to read our network file. So once we read and just convert like in a matrix, and it can check the matrix how much dimension. Yeah, we have to remove second one because we have to remove interact that word. So like this is v2. We have to remove v2. So just we did this mat like now dimension become 2. Now we have to calculate the graph edge list and make a matrix like if A is interact with A, it appear like 1. If not A interact with B, it means like 1 0 1 0 2D like a binary matrix we have to create. That is called adjacency matrix. So it will like take little while to make a matrix. This method is good for if you didn't know any like biomarker hub genes. So what you can do like in a transcriptomic data, you just take all the differentially expressed proteins and make a protein-protein interaction network, and now you want to know which protein is more which protein is just lead as a biomarker in all these differentially expressed proteins. So we can use this method, just make a protein-protein interaction network and just go by community finding method and go to break the network up to motif level. So you can get at the end few marker protein which are present from top to bottom. So that means these proteins are very important to form and stability uh, to form the network and make it stable. So so like we have make a matrix now.
So these are triangle motifs, no?
Yeah, adjacency yeah, triangle motif.
Okay.
Sometimes triangle motif, sometimes like clicks, and sometimes like rich club, also called
Yes,
rich
yes.
club. So all these are basically looks like similar term, but it depends on.
Yes.
So now we are checking how many. Now we can break this matrix into how many communities can be break. So now we can check the size. So suppose our network will break in 4 communities. First communities contain 924 nodes. Similarly, all these 4 communities contain this number of nodes in their community. So in a first level, this is the first level.
Okay.
Like 0 level, we have just take our main SIF file. So now it is the first level. Now these 4 communities also like here, like now these are the 4
Yes.
communities. Now we will go this one, but here we have to finish it. So now we have to extract these communities. So like we have to take 4. If like you can change here, like if like 5.
Okay.
In our case, there are only 4 community detected, so we just take 4. Otherwise, we can take how many number of proteins. So we have just selected 4. So write these communities over it. Again, we can save these communities with your name. Like I save it like network 1, 1.1, 1.2, 1.3, because like main network as assigned as a 1, and that is divided into 4. So 1.2, 1.2, 1.3 like that. So here you have to be more careful about in the numbering system, because when you break, then just in a hierarchical manner you have to assign a number, because in the end when we predict all this community, then it is easy to analyze the communities.
Okay.
So now we run all these four communities are in our folder. Now we can check like this, like this. So now what we can do, just select all these protein and search here. Like that. Oh, sorry, because we have to remove mark columns. So 924 proteins we can just make another network. This is the first community. Just rename as 1.1. Okay. So similarly, suppose I'm skip both these and take into remove all these like inverted commas and commas. Otherwise, not.
So it doesn't work with the commas.
Yeah, I removed because comma not detected in Cytoscape. So
okay, so we should pay attention and print them
Yeah,
from R without any commas.
yeah. So no. So here we can extract like selected node all edges. So this is 1.4. So when you're doing so simultaneously you just rename all the things so you will not mess up. So these are the two communities. So now if you once we this is first level complete, suppose now we are going to second level. So what we say, just save it as a network file. Okay, this file should be saved in a network file like SIF file. So network file, this is SIF file. So 1.1. Okay, save it. Network file. I think. Now again we like recite means we can again run the same script by using like 1.4. So now first level is complete. Now we are going for second level. So first in second level again we can read all this following that's all these step. Now this 1.1 is break into 5 communities.
Okay.
So now we have to in the second level break. Now 1.1.1. Now we can save again. So this type. So similarly, we have to just one thing you have to notice that there is a modularity score. Is showing. Yeah, this is not valid. So you have to go for in this level for plotting. So for the like we have to repeat this script till the like network is unbreakable. So it will show 0, like community is like showing 1 2 3 4. So when like if you found like suppose one network is broken in 3 communities and each communities contain like 5 6 7 nodes. So I think it's my suggestion you have to be stopped there, because in 5 6 community you can easily detect what are these communities, what are the these communities contain our interest of protein or not. If like these one more thing important thing I have to tell you, like you suppose, yeah, I have to draw, I think. Suppose like we have like find all these communities. Now we have to check the quality of the communities. Suppose one community is like this triangle community, that means that is a motif, we have to keep it. Otherwise some communities like that in a linear like this, so we have to discard these communities because it is a meaningless. In a graph theory, if communities are interacting to each other, that means these are the meaningful. But if it is like in that manner, we should remove. But if it is like this again connecting, we can consider. So similarly, many proteins are like that, we can consider. So if it is like interacting to each other, then we can consider. So once we have like all these SIF file, just open it and check, and which are not useful, then discarded it. So at the end,
So,
yeah.
sorry, just a question here. Should it be like any such a community? Should it be a complete graph, or are there any other? So, for instance, the one on the right hand side, the right handmost is not connected with the one in the lower part.
So, but it is connected with this all these three ones, so we can consider it is. But if it is not connected with this, then we can reject.
Okay, so it should be a complete graph, right?
Yeah, complete graph for
Okay.
functional values. We have to see these communities and subgraphs should be complete. So not necessarily like it is, if it is like many networks are not unbreakable. Like here when you break this community, it shows like 0 communities. So we have to consider this one. We can't break it.
Okay.
Because it calculates topological properties. If like all the topological properties are like or valid, then we can't break in these communities. So we have to just leave it and consider is as it is. That sometimes that community contain 20 nodes, 25 nodes, but it is unbreakable. That means that community is working with together with along with all these nodes member.
So
yeah,
here on the right side,
yeah,
it's not complete. I think that it should be considered because it has a motif, and the others are connected to the motif. No.
Yeah. This is called connected with motif. So we can
okay, we can keep it. Yes.
Yeah, we can keep it. Yeah.
Okay. Yes. So you check for some motifs, and everything which is connected to the motifs is kept.
Yeah. If it's something connected, we can keep it because functional graph, functional properties of these are performing similar function. But maybe it is regulate these proteins, so we can keep it.
Okay. Yes. So you find you should check for several types of motifs, clicks or triangles or so on, and everything which is connected to the motifs is kept.
Yeah, exactly. So first we have to run all these script. Then here you can check all these ones. All these levels are here. You can check one by one. So it's my suggestion. If you broke one level, just make it in the like in the Cytoscape. In the second level, in the Cytoscape. Don't wait file. Like at the end, we will go and then plot. Maybe some confusion we can create. So if I find time, so I will try to write this script to combine all these, not going by one. So
together.
Like once run script and extract from PPI network and go again. So it's going loop. But this script should be loop. If in future, if I found time, then I will write some function. So suppose there we have to write one by one. We have to doing now. So what we do, like this subgraph is saved. So what we will do, this subgraph take protein from subgraph and search in the main network and extract them as a SIF file. Then SIF file read as a input file and again run this script.
Yes.
So like that is a loop. So so it is easy to
No, it's okay.
Yeah, so it will be easy for us, like not going by one because some networks are too big. So like we have to give break community 6, 7 level, and each level has like 20, 30 like that. So it is a very time taken process. But it's good actually. If you don't know the biomarker and anything, then you can find from this network. It is very good.
Yes. Yes, it's very nice.
Okay, so we can go next. Yeah, Mtgo, we can go now. So it is like you understood all the things. Any confusion you can ask.
I think so. I think that's okay.
Okay, so
I think it's fine. It's quite structured and it's understandable. And even for the next iteration, it's great.
Yeah. So now it is like Mtgo. That is also good, and I'm convinced with this result. So like suppose we have like known proteins target, then we can use DIAMOnD and Mtgo. That is good, give the good result. If you like, we don't know what are the biomarkers, what are the key functional regulator in our bunch of proteins, then we can use like Levy method. So here input first we check the input files, like input file as like protein tab and one protein. So interactor 1 tab, interactor 2. Here is a one tab required. This is our network file. Now this is our all these.
I have a question.
Yeah.
I want to ask it now in order not to forget. Both in this example and the former example, you already have a network of edges that you're interested in.
Yeah.
How is that one picked?
You can pick, like suppose you have like 50 proteins, just go and make network from string, biogrid, any database you can create network. So I can show you even. Okay, I will, like here you want to make a network, suppose. These are import network. You can from file, public database. If you like little, like a few 50 protein, 60 protein, then you can use like a public data. network from public database, this one. So you can paste all the proteins, like these are the wiki pathway and string protein query. So here you can get two pathway, but like also you can select from here. If like you select universal interaction database client, you just, I will show you. I will select some proteins to show you network, how to construct network. Suppose I select random proteins, these protein 54. So you can select databases from which database you want to make. So generally we select IntAg, IntDB database, primary database we generally selected, Reactome, Mint, all the secondary database. So we can select any database. And this is the option automatic network merge. So predicted from these databases, these network merge. But I will not suggest to do it by doing this. By my personal suggestion, just go in a BioGrid and Sting databases and download all the network, whole proteome interactome, like this. You can get you can save in a SIF file like this. So my personal like that, I will show you again. Last time I will share you how to download.
Yeah, I see. So, yeah.
Like that tab 2 file. I will suggest you.
Okay, I understand. Thank you. Thank you very much.
So it is a good thing, like you can get the maximum interaction, because in the doing that, if you are like a smaller number of protein, like 20, 50 protein, then you can use this Sting and IntAg database. If you have a huge number of protein, then you downloaded all the databases, all the proteins from databases in Cytoscape and search our proteins and extract as a secondary network from primary interactome. So I will start this Mtgo. So this is the like Java platform here. So just you have to select your edges and ontologies. You provide edge file here. So here we can select minimum cluster size. So cluster size generally we take from like 3, because like at least some motif level we can select. So in a 3 nodes should be in a one cluster. That is the minimum criteria. And maximum geo size is just like how many this cluster present in at least minimum 30. So here 30 to 300 size it takes. So we just take a basic initial cutoff values. So we can not we can so doing this we can extract maximum information from this. So later we can remove like on the basis of Mtgo values and p values. So now it will take time, at least it will depend on your network file. So in meanwhile I will share some. So now I will show you like MCODE, molecular complex detection method. That is another method that detect the mostly dense region in the protein detection network. So we can download ClusterViz. So there is ClusterViz app in a Cytoscape. So what it did, we can select MCODE. Here you can set degree threshold. Degree threshold means like how a node connected with the how many number of nodes. So we can select at least 3. And K score is like a minimal degree of K. So that means highest K score of graph. That means that the node is a central node and most densely connected subgraph. So also take 3 degree maximum depth. Okay, so we selected this network for finding clusters. Sorry, I will directly. I think this network is not feasible because no, it is not a cluster mode. This is just like a one node has a multiple degrees. That's why it's going error. I will select some other. For this. Sorry, what happened? I just restart. It is running, still running. It will take time. I'm forgetting something here. What happened? Okay. Once you start, just read this about like what haircut fluff, because in haircut and fluff very little bit different. It is like if one node is interact with one or two nodes, so we have to take on node, and what is the overall topological value, some kind of, these are the theory they have given. I read, but I forgot, I think. So once you restart, then just read it about little bit. Oh, still running it is. Yeah, sometimes it took time. Network edges is too much. Now we started this calculation. So I will show you now old result, which is I have run in that somehow, because it may take more time. Like these 4 file it will generated after running this query. So first file is like this one. So it shows the number of proteins involved in the cluster wise. Like cluster 1 is like number of if you open it in the like that. And second, these 3 file can be used in the Cytoscape to make a. These are the score. How this at time iterate and give the modularity score for each run. That will also showing here one by one in the screen. Like 81 clusters it has predicted, and each clusters performing the same similar kind of functional and notation. That is like one kind of over representation analysis. Suppose this module perform this specific function, this module perform a specific function. So these 2 modules are like cross validated. First is like node module 5. That means these modules are verified by like here you have to remember that Mtgo just predict by 2 methodology. First is like topological properties, and second one is like ontology terms. So these are the 2 things. Module 5 is just like topological. It takes from topological value, and this is predicted by like geo term. So mostly both the list of proteins mostly similar in, but in a topological properties little bit more proteins. So but why I am suggesting that if you are using to perform doing pathway enrichment analysis or other analysis, you have to take Mtgo result, because it predicted by 2 methods. So it is already cross validated. So the chances of the perfection of this module is much higher than other methods. So that's why, because you can also check how many proteins are there. Yeah.
For the code and for the code snippets and for the data, can you please zip?
Yeah, I will zip all the file and I will send you. So like this is the smallest code to count the number of items in the column. These are the time taking little bit. So these are the total proteins involved in all these cluster. It is running still. Can I like cut this because both are taking CPU's power? So
Yes.
it become
Yes, okay.
because it is very easy. You can. I will send you all these things.
Okay. Oh.
I will see. Take a smaller one. In a previous meeting, all the things are clear. So like
Yes.
this.
For my part, at least.
Okay, so you people will come to USA, and Dr. Kiosi told me.
Yeah, in
Yes.
2 weeks and 2 days.
18.
Okay.
18th.
Okay.
We will meet there. Yes.
yeah, definitely. So like the MCODE predicts this number of cluster in the module. So it predicts on the basis of like how densely region. So we can take as it is, or we can again go save this one, like this one, like 1.1, any format, and then again go for like level 1, level 2, level 3. So mostly people do what we do, they just take cluster like this, cluster 1, cluster 2. All the cluster, important thing that your protein is available or not. Suppose in this cluster, if your protein is not available but other interactant proteins are available, then we don't need this cluster, we can discard. Because we are when we preparing like constructing protein interaction network, in that network our list of proteins, suppose 100 200 proteins, but other interacting proteins are also present. So sometimes what the these methods, all these clustering method predict the proteins without our query protein, so we can discard this cluster because that cluster is useless. Suppose in this cluster 3 4 5 proteins are our query protein, that cluster become more useful for us, then we can go for functional analysis and clustering pathway analysis. So these are the methods, very easy, and you can perform all these clustering I think from my side I'm done. Any question you have?
Okay, so yes, I think that's very useful. Okay.
Yeah. Also, if you have like network, you can go for like topological properties analysis, like here, analyze network. Yeah. From here, you can also get the which are the proteins has a number of closeness centrality, clustering coefficient, all these details we can get. And also we can get which protein has a number of degree. The number of degree that protein play important role in the network by interacting with other different proteins. Like suppose here HSP8 is interacting with approx 114 proteins, so that is important for us. And that protein, that higher degree protein in the network reach to up to the bottom level. Like we are going by level 1, level 2, level 3 now. These proteins will reach to down level because these are the core protein which are present and make a stable network. If we delete this protein, many proteins are become like orphan. Like that. So these core proteins are higher in degree, higher in closeness centrality, higher in all these topological properties.
And somehow these proteins make this great degree, with high degree, make some hubs. So they make small worlds there.
Yeah, they are a small word network actually. If you're talking about like degree, like one HSP8 is like connecting with 115 protein. That means that is a small word network inside the main network.
Yes.
Yeah. So many useful information we can get by topological properties. We can plot and fit the linear modeling. Many mathematics we can do.
Yes. We will do.
I will share one paper you can follow. Like I think that is good paper.
Yes, please.
Yeah, I will share by using these topological properties. So automatic, I will share these few papers. They're using mathematical some formulas, and then they created like some graph plot. So and one more thing, like knockdown experiment they have done, like here network. Suppose we have delete some protein, like important protein we deleted from here. So what is the effect after deleting by this protein? So like suppose we have in our suppose we have find 10 biomarker at the end of the community finding algorithm. At the end, in motif level, we find 10 proteins. That is most probable. Like we can say like these are the key regulators. Okay. So what we do, like we deleted from main network each protein. And check the effect. What are the effect after deleting by this protein? This protein. So one by one we can delete protein and check the effect through which protein most affect the network stability. So that protein may be more important for the network. If we target this protein, that network may be collapse. So in terms of this type of study,
maybe they break. They can break the network.
Yeah, collapse. Yeah.
Something like this. Yes. Okay. I understand. I see.
Yes.
Yes.
It is kind of a study. We also do that.
So maybe are not only high degree proteins. For example, if you can imagine that you have two separate networks.
Yeah.
And they are connected only through a node.
Yeah.
So if you break this node, that node, which has no high degree, in fact, you can break the whole network.
Yeah, maybe.
So somehow there are other important nodes which somehow realize some connection between some clusters or something like this.
Yeah, we can do that.
Maybe as I see now when we talk so first time.
Yeah, we can do that. Many things like if you like if you're talking about like shortest path, suppose. So in the shortest path, we can get the maximum number of protein which is more near to particular protein that form a complex. So suppose in a graph theory, it is concept that if one protein is interacted with the in one protein is interacted with by like 3-4 different proteins, so that means that protein is also involved in a similar function. So in a graph theory, that is called like guilt by association. That is a term, guilt by association. So we can also get the many like proteins are we don't know, but when we find the cluster and when extract our interest of module or motif cluster anything, then we also extract that particular protein that is unknown. But when we like analyze the result, then we find that these proteins play important role by regulating, by inhibiting our protein of course, our interest of protein. So that protein also is important. So that proteins we call like inferred protein basically. So we can get the more other protein also along with our key regulators.
Okay.
So many information we can get from network theory. So it's depend on how you are analyzing the data, what you want from the network, what your question, what your demand from the network. Like
yes. So please send me if you have some papers.
Yeah, please. I will send it.
Okay, please.
Okay. So anything else for next? I think I'm done. Anything else if we can schedule?
Okay. And so you can zip also for us the files and.
Yeah, today fine. No? I will send all this data. Yeah.
Okay, please.
Yeah.
Very nice. Thanks. Very, very interesting. Yes.
So Faith, come and see you here. Hope so.
Okay. Yes.
Yeah.
Thank you for the presentation.
Thank you. Very nice.
Okay. Thank you.
So next week.
Next week, if you have any query, then maybe we can meet. Otherwise, I think I am done.